Hello everyone. In today's lecture, we will talk about affinity chromatography. That's a very powerful technique for purification of analytes, including biomolecules. In earlier lecture, we discussed about basic principle of chromatography, and we understand that in a chromatographic system, you must have a stationary phase and a mobile phase. The function of a stationary phase is to interact with the analytes, and basically the separation of analytes in a chromatography system occurs because of differential interaction, a different binding of analytes to the stationary phase and the mobile phase. So in a column chromatography, you take a column and you pack a stationary phase in the column, and you load on the top of the column, you load the mixture of analytes. Now these analytes are pushed into the stationary phase with the help of a mobile phase. So mobile phase take these analytes and it goes to the stationary phase so that analytes can interact with the stationary phase. So based on these interaction, the analyte which interact strongly, which binds strongly to the stationary phase, that is retained back, that moves slowly in the column when you are continuously pushing mobile phase from the top, and analyte which does not interact much, which make weaker bond with the stationary phase, that move faster. And that's how these analytes are separated in a typical chromatography setup. Affinity chromatography used reversible biological interactions between components in the mobile phase and the solid stationary phase. So here you immobilized ligand on the stationary phase. This is a solid bead which forms part of the stationary phase. So in the solid bead, it's called matrix. So on the matrix you immobilize ligand, and this ligand should have a very strong specific affinity for a particular analyte. You can use affinity chromatography only to the condition when you have a specific ligand available for the particular analyte you want to purify. For example, if you want to purify a protein, an enzyme, and you have inhibitor available for that enzyme, that inhibitor can be immobilized on the stationary phase, and that can be used to separate that enzyme in affinity chromatography. This process, this interaction between the ligand and the particular enzyme is highly selective and highly specific, and that's why generally affinity chromatography makes a single step purification of biomolecules. It uses like a lock and key mechanism. For example, you have a substrate or inhibitor to the enzyme, and this is the enzyme which have interaction with this. So this substrate can be immobilized to the stationary phase, and that can be used as a matrix in order to purify. The interaction between ligand and the particular analyte, those are biological interactions. Those are mainly electrostatic bonds, hydrophobic interactions, or van der Waals forces, which involve between ligand and the interaction between ligand and the particular enzyme or analyte. The nature of stationary phase varies in affinity chromatography. That's how you have different type of affinity chromatography. For example, if you are interested to purify an enzyme, you can use inhibitor, cofactor, or substrate of that particular enzyme so that that enzyme can be purified from a complex mixture. Similarly, if antibodies are available for the analyte to be purified, you can use antibody and you can mobilize those antibody into solid stationary phase, and that can be used for purification. Similarly, if you want to purify glycoproteins or polysaccharides or cell surface receptors, lactin can be used in stationary phase. Lactins are a family of protein which binds to sugars, so sugar binding proteins that can be immobilized to solid matrix in order to purify polysaccharides and glycoproteins. Similarly, if you are interested into fatty acid binding protein, fatty acids can be immobilized on the stationary phase, or hormones, vitamins also can be immobilized in order to purify any analyte which can interact with them specifically and strongly. Similarly, is very commonly used glutathione is a tripeptide that is used in order to purify glutathione as transferase GST or GST tagged fusion proteins. Very commonly used method in order to purify proteins in molecular biology laboratories. Or even metal ions can be used in stationary phase. metal ions interact with the histidine and cysteine amino acids in the protein. So that's how metal ions, that forms basically metal affinity chromatography, another variation of this affinity chromatography. So metal ions can be used to purify histidine containing proteins or phosphoproteins. In order to purify antibodies, protein A and protein G are used in stationary phase. Protein A and protein G, they are bacterial proteins which bind to Fc region of antibodies. So if you want to purify antibody, these are the affinity column must have either protein A or protein G immobilized in the stationary phase. Similarly, phenyl boronate is used for glycoprotein purification. Poly A tail can be used for RNA containing poly-U sequence or RNA specific proteins purification. And nucleotides also can be immobilized in the stationary phase in order to purify nucleotide binding proteins. So there are 3 or 4 steps in affinity chromatography. The first step is to prepare matrix, to prepare a stationary phase, where you immobilize ligands into some solid matrix, solid surface, and then you load the mixture of analytes on the top of this column. You pack these immobilized ligands. The stationary phase you pack in a column, then you load this mixture of analytes, and when you push the mixture of analyte with the help of a mobile phase, most of the analyte which do not interact, they flow through the column. They pass through the column, and you can detect them here in the chromatogram. You see a very big peak initially that represent unbound fraction, the analyte which does not interact with the stationary phase, and then the binding occurs between the specific analyte and the ligand immobilized in the stationary phase. Now, in order to remove those analytes which is interacting with the stationary phase with the immobilized ligand, you can change composition of the mobile phase. Either you change pH or change the ionic strength, and very commonly used way to remove this interaction between ligand and the bound analyte is to use competitive molecules. So you can use competitive molecules. When you use competitive molecules, these molecules bind themselves to the stationary phase, and that's how the bound analyte that's removed from the column and that get eluted into form of another peak, which is generally a pure analyte. This process is called elution. Principle, there are 3 components of affinity chromatography. Is specificity between ligand and the analyte under question which you want to purify. The ligand should be specific. If it's not specific, it cannot be used in affinity column, and hence it will be of no use in affinity setup, affinity chromatography. Then the binding should be reversible in nature. So once the ligand is bound to the analyte, that interaction can be disturbed and can be removed, so that bound analyte can be eluted out from the column. And of course, you have a stationary phase. A stationary phase is composed of a matrix where the ligands are covalently immobilized. That's used in affinity column. So they are in fact a 3 component of a affinity column. One is matrix. Matrix is the solid stationary phase, generally a polysaccharide. It could be agarose, polyacrylamide, polystyrene, cellulose, etc. So on this solid support, you immobilized a ligand. This, for example, the nickel is the ligand here, which is immobilized, and this matrix should be chemically, physically inert, so it does not interact at all with other analytes in the mixture, and it should provide high surface area for better ligand attachment. And of course, it should be insoluble in solvent and buffers which you are going to use in mobile phase. The second component of a column, affinity column, is spacer arm, or that's called linker. The purpose of the linker is basically a chemical group which connect ligand. For example, this nickel ligand is connected to the bead through this linker. So this chemical group is called linker here. So the purpose of linker is to connect solid support, the agarose or polysaccharide beads, to the ligand. This is required in order to expose ligand outside, so that this ligand can easily interact with the protein or analyte under question. So this linker, it connect ligand to the matrix. It improves binding efficiency between target molecule and the ligand by overcoming steric hindrances. So there is no steric hindrance. There's a purpose of a linker to expose ligand on the surface in the mobile phase, so that analyte under question, the analyte which has specific binding to the ligand can interact easily. Then the third part is a ligand itself. The ligand refer to the molecule attached or immobilized to the matrix, which reversibly bind to a specific target. So this ligand could be a metal ion if you are using metal affinity chromatography. It could be antibody if you are using immunoaffinity chromatography, or so on. This ligand varies depending on what kind of analytes you want to purify. In fact, the ligand could be of two types. The monospecific low molecular weight ligand. Monospecific means which are highly specific, for example, enzyme inhibitors or steroid hormones. Enzyme inhibitors, the inhibitor of any enzyme is very specific to that given enzyme. So if you are using some enzyme inhibitor, the specificity is very high, so you can easily purify the target enzyme using that. Even another ligands are group specific ligands, which bind to a family or category of analytes. For example, nucleotides. If you immobilize nucleotide in the stationary phase and you are intended to do nucleotide affinity chromatography, in that case, a group of protein, group of analyte which can interact with the nucleotide, all will be retained. So those kind of ligands are group specific ligands. Applications you use in fact affinity chromatography for purification of proteins or purification of antibodies, or in fact purification of any other biomolecule or analytes can be used, or cell separation. You can purify cells also. The only requirement is you must have a ligand which can specifically interact with the given analyte. If you have that, you can use affinity chromatography, and most likely you are going to have a single step purified analyte using this technique. The factors affecting affinity chromatography is specificity of the ligand. So that is the prime importance. You must have a ligand which is highly specific. Only then you can use it. Otherwise, you can't do that. You cannot use affinity chromatography. Then the affinity of binding strength of the ligand. The binding strength of ligand should be optimum with the analyte. If it is too low, then most of the analyte will not bind to the column. Your yield will be low. And if affinity is too high, it will be very difficult to elute the bound analyte from the ligand. In that case also, the yield will be less. So you must have a ligand with optimum affinity with the analyte, so that it can be used in an affinity chromatography matrix. The nature of the matrix must be inert, and generally it should not interact with any other analyte. For example, say if you are using agarose beads. So if you have in the mixture of proteins, you are purifying proteins, and you have some protein which can bind to sugar. So those may bind to the agarose, and that's how you may have another step to purify. It may not be single step purification. Then your pH of the mobile phase should be optimum, so that binding between ligand and analyte can be optimized. The nature of the buffer used, the buffer should maintain the pH. It should have a proper ionic strength, so that ligand and target molecule, ligand and analyte, they are happy, they are stable there. And then when you are using chromatography system, your flow rate should be optimal, should not be too high or too low, so that your analyte is not purified properly. Advantages. The advantage of affinity chromatography is the one that even using is just single step, you can purify your analyte under question, and that is because of high specificity. The interaction between ligand and the analyte is highly specific, and that's why it forms a single step purification of the analyte. It has high purification factor. Purity is generally very high when you are using affinity chromatography, and molecules can be obtained in highly purified form in high yield. Yield also is good because you are using ligands which is strongly and specifically interact with a particular analyte only. The matrix can be reused. That's generally in all chromatography setup. Effective purification based on biological structure and function. Your recovery of the compound is high. That's why yield is high in affinity chromatography. And independent of the sample volume provided, the ligand is highly specific. So it doesn't matter how much volume you are loading on the column, because here your molecules will be interacting with the ligand, and that's how Doesn't matter the volume, initial volume of the analyte you have. Limitations: it's a time consuming and complicated method, and limited availability and high cost ligand. This is the major limitation. In fact, the ligands are not, if it's not available, you cannot use it, and generally they are costly ligands. So when you are using, procuring them, they are expensive one. It's a labor intensive procedure, and non-specific adsorption cannot be totally eliminated. Some other molecules also may bind to the matrix. That's what generally happens in affinity chromatography. And proteins get denatured if the required pH is not adjusted. In fact, this is common to other chromatography techniques also. If pH is not proper and you are purifying proteins, in that way you need to get the best optimum pH so that protein is stable and optimum interaction between protein and ligand can occur. Now the types of affinity chromatography, depending on the matrix you are using, there are variations in affinity chromatography. For example, you can have boronate, phenyl boronate affinity chromatography in order to purify glycoproteins, or hemoglobin HbA1c, that is purified using boronate and phenyl boronate affinity chromatography, or lactin affinity chromatography, where lactin is immobilized in stationary phase, and that's used to purify polysaccharides or glycoproteins. And you may use dyes also, because there are dyes which bind to several proteins. So if you use dye, it's called dye ligand affinity chromatography. Or, and if antibodies are available, you can immobilize antibody in stationary phase, that's a immunoaffinity chromatography. Or even if you have metal ions immobilized in the stationary phase, all the metal binding proteins can be purified, and that's called immobilized metal ion affinity chromatography, IMAC, or just metal affinity chromatography, that we will discuss in more detail in the next lecture.